Did modern life on Earth evolve over millions of years, or was it created in the blink of an eye by God? This is a binary choice that most humans have accepted since the mid-19th century. And before that, there was simply the single explanation: God created man. But for the last 160 years, a battle has been waged between the scientist and the creationist. The scientific evidence is clear. The Earth is about 4.5 billion years old, and all life evolved from primitive, single-celled organisms. Nevertheless, biblical literalists reject this evidence and operate from the viewpoint that the Bible's book of Genesis is a historical account of creation. Perhaps the most fundamental tenet of the ancient astronaut hypothesis is that it offers a third option, an option that is based on the scientific principle of evolution and the oldest stories known to humans about our creation. It is in the ancient Sumerian text that we actually find accounts that combine evolution and creationism, stories of an ancient race known as the Anunnaki. Throughout the millennia, world religions and literature have possessed a recurring theme of the creation of man from clay. Interestingly, the role of clay minerals in the origin of life can be suggested even scientifically because of the ordered arrangement of the clay mineral particles, the large absorption capacity, shielding against ultraviolet radiation, ability to concentrate organic chemicals, and ability to serve as polymerization templates. Also, clay minerals act as very efficient catalysts in the polymerization of amino acids and nucleotides, thus supporting the possible existence of an RNA world preceding a cellular world. So, ironically, the scientific data obtained so far suggests that clay minerals played an active role in the abiotic origin of life as it pertains to Darwin's concept of evolution. Here are some examples of the creation accounts throughout the world. In Greek mythology, according to Apollodorus in the Bibliotheca 1.7.1, Prometheus molded men out of water and earth. According to Egyptian mythology, the god Khnum creates human children from clay before placing them into their mother's wombs. According to Chinese mythology, Nuwa molded figures from the yellow earth, giving them life and the ability to bear children. According to Hindu mythology, the mother of Ganesh, Parvati, made Ganesh from clay and turned the clay into flesh and blood. According to some Laotian folk religion, there are accounts of humans created from mud or clay. The Yoruba culture holds that the god Obatala likewise created the human race from clay. According to the Quran, God created man from clay. The Maori people believe that Tain Mahuta, god of the forest, created the first woman out of clay and breathed life into her. According to Inca mythology, the creator god Viracocha formed humans from clay on his second attempt at creating living creatures. According to some Native American beliefs, the Earth Maker formed the figure of many men and women, which he dried in the sun and into which he breathed life. In Norse culture, humans are made from sand in tree trunks. Perhaps the most famous account is in the Bible. According to Genesis 2:7, quote, "And the Lord God formed man of the dust of the ground, and breathed into his nostrils the breath of life, and man became a living soul." End quote. In the Babylonian creation epic Enuma Elish, the goddess Ninhursag created humans from clay. And in the Epic of Gilgamesh, Enkidu is created by the goddess Aruru out of clay to be a partner for Gilgamesh. Thousands of years before the Bible came into existence with its version of the creation of humans, the Sumerian text, in much greater detail, chronicled the Anunna's genetic experiments as they engineered the first humans. They explain it took 6 attempts to reach a successful outcome, which would allow for the insemination of a new humanoid to produce the first true humans. Nimma was the original scientist trying to engineer humans. The electronic text corpus of Sumerian literature states in entry 56-61, quote, Ninmah took clay from the top of the Abzu in her hand, and she fashioned from it first a man, end quote. But after multiple failures, she became frustrated and asked Enki for assistance. Many researchers have speculated this genetic engineering took place in what the Bible calls the Garden of Eden. They posit that Adam and Eve were the first two creations and were used to procreate humanity. Indeed, the garden could have been a biosphere, one where the genetically engineered humanoids were studied in order to determine any needed improvements. In any case, the Genesis story of the creation of man is seemingly rooted in Sumerian history and the Anunnaki. It is also important to note that the reference cited here, known as the electronic text corpus of Sumerian literature, is the same tool academia utilizes for accepted transliterated Sumerian text. It is not an interpretation by an amateur researcher or author. Of course, academia dismisses such accounts as myth, and therefore any serious inquiry into the potential scientific feasibility of such accounts is virtually non-existent. The entire set of entries from the Enki and Ninmah translation found in the ETCSL have been posted to the archive website. A link to that page and a direct link to the corpus have been provided in the description. Ever since Charles Darwin published The Origin of Species in 1859, the general public has come to believe that he is the father of the theory of evolution. Yet, he never said that man descended from the monkey, nor did he even invent evolution. What Darwin did was construct a whole theory from an idea which had been floating in the scientific ether for a century, ever since Carl Linnaeus began to classify the species by their similarities and left behind a diagram with many branches that emerged from a few trunks. Seeing that family tree, it appeared that the species were related and had evolved from common ancestors. Interestingly, Darwin's concept of survival of the fittest derived from the economist Malthus, who, at the start of the Industrial Revolution, and with the rising problem of urban overpopulation, thought that many people were being born that could not survive due to the lack of food and space. Darwin applied that idea to nature. Creatures multiply, and in struggle to survive and reproduce, those that flourish are the ones that are better adapted, that are born with some advantage. Their offspring inherit the trait, and little by little it becomes more common. And they continue to thrive and change for thousands of generations, ending up as a different species. However, despite the name of his book, Darwin never explained the origin of the species. He did not understand what it is that drives the motor of evolution. Modern science argues that the cause is genetic mutations. It posits that human evolution is the evolutionary process that led to the emergence of anatomically modern humans, beginning with the evolutionary history of primates, in particular genus Homo, and leading to the emergence of Homo sapiens as a distinct species of the hominid family, the great apes. This process involved the gradual development of traits such as human bipedalism and language, as well as interbreeding with other hominins, which indicate that human evolution was not linear, but a web. The study of human evolution involves several scientific disciplines, including physical anthropology, primatology, archaeology, paleontology, neurobiology, ethology, linguistics, evolutionary psychology, embryology, and genetics. The evidence on which science relies for human evolution has traditionally been the fossil record, but since the development of genetics beginning in the 1970s, DNA analysis has come to occupy a place of comparable importance. The studies of ontogeny, phylogeny, and especially evolutionary developmental biology of both vertebrates and invertebrates offer considerable insight into the evolution of all life, including how humans evolved. The closest living relatives of humans are bonobos and chimpanzees and gorillas. With the sequencing of both the human and chimpanzee genome, as of 2012 estimates of the similarity between their DNA sequences range between 95% and 99%. But using the technique called the molecular clock, which estimates the time required for the number of divergent mutations to accumulate between two lineages, the approximate date for the split between lineages can be calculated. The gibbons and then the orangutans were the first groups to split from the line leading to the hominids, including humans, followed by gorillas, and ultimately by the chimpanzees. The splitting date between hominin and chimpanzee lineages is placed by some between 4 to 8 million years ago, that is, during the late Miocene. Each time a certain mutation or single-nucleotide polymorphism appears in an individual and is passed on to his or her descendants, a haplogroup is formed, including all of the descendants of the individual who will also carry that mutation. By comparing mitochondrial DNA, which is inherited only from the mother, geneticists have concluded that the last female common ancestor whose genetic marker is found in all modern humans, the so-called mitochondrial Eve, must have lived around 200,000 years ago. For years, the consensus has been that Homo sapiens emerged from one spot and one population in East Africa some 300,000 years ago. But according to a new paper in 2018 in the journal Trends in Ecology and Evolution, we didn't come from one place or population on the continent. Instead, lineage of Homo sapiens probably originated in Africa at least 500,000 years ago, evolving from interlinked groups across the entire swath of Africa toward contemporary human morphology over tens of thousands of years. This expansive change was influenced by ecological and genetic shifts, according to the authors. So, here is the big question at this point. What if those genetic shifts were not the result of natural evolution? What if the mutations were the result of external intervention by an extraterrestrial source? The signature for genetic engineering of our hominin ancestors is a complex subject because of the considerable number of modifications to different parts of the genome. If an advanced alien race such as the Anunnaki visited ancient Earth and decided to modify an organism like our hominin ancestor, it would likely be extremely difficult for us to detect this event. Nevertheless, if extraterrestrials intended to leave traces of their genetic manipulation, it would certainly be possible. Any such signature would simply be encoded in regions of the genome that are stable and become highly conserved. Indeed, some strips of DNA letters have remained the same over millions of years. Chromosome 2 is one of the 23 pairs of chromosomes in humans. People normally have 2 copies of this chromosome. It is the second largest human chromosome, spanning more than 242 million base pairs and representing almost 8% of the total DNA in human cells. While humans have only 23 pairs of chromosomes, all other extant members of the hominin have 24 pairs. Science argues that chromosome 2 is a result of an end-to-end fusion of 2 ancestral chromosomes. The closest human relative, the chimpanzee, has nearly identical DNA sequences to human chromosome 2, but they are found in 2 separate chromosomes. The same is true of the more distant gorilla and orangutan. Mainstream science claims the second and third primate chromosomes were somehow accidentally fused at some point in time to form a single chromosome. However, creationists say it makes more sense that humans were created this way by an intelligent designer, and some deny that the chromosome is even fused. The assumption in both groups points back to the binary choice we spoke of at the beginning of the presentation. Basically, it had to be either Darwinian forces or God. A couple of other differences between us and primates are duplicons and certain micro-RNAs. Humans and primates have a duplicating function in their genes present in no other animals, which are called core duplicons. Although they can be found in primates, they are present in far greater numbers in humans. These duplicons tend to be focused on genes that enhance brain size and function. The recent discovery of several micro-RNAs, such as MIR941, has brought us an example of a gene completely unique to only humans. The gene is highly active in two areas of the brain that control our decision-making and language abilities, and it is believed to have a role in the advanced brain functions that make us human. So now that science has made these new discoveries, it has brought about a new set of questions. Why are there all these specific, uniquely human genes related to brain function? Do these new discoveries bolster the arguments of science or creationists, or possibly give credence to the idea that some other intervention took place? The scientists and creationists both have their issues with these new discoveries. Science has the problem of explaining these rapid mutations, and creationists have the problem of their God looking a bit sloppy by leaving evidence of spliced primate chromosomes in his human creation. For those who consider the third option of intervention by extraterrestrial sources, another segment of the human genome found on the long arm of chromosome 20 is of particular interest. It's called Human Accelerated Region 1, or HAR1. One of the more common comparisons used to illustrate the anomalous nature of this mutation is the one between a chicken and a chimpanzee. There are regions of DNA code which are almost identical in the genome of both species. One of these overlapping segments of code is 118 letters long and differs by only 2 letters between these species, which are separated by 300 million years of divergent evolution. In contrast, in the same code segment compared with humans, we find an 18-letter divergence, despite chimps being our closest living relative and diverged by a mere 6 million years. This is another problematic issue for science because its current model indicates that an 18-letter divergence is far more than expected by the region's history of conservation. Biostatistician Catherine Pollard stated, quote, "The fact that HAR1 was essentially frozen in time through hundreds of millions of years indicates that it does something very important. That it then underwent abrupt revision in humans suggests that this function was significantly modified in our lineage. Statistically speaking, the probability that a highly conserved DNA sequence will change multiple times over 6 million years of evolution is close to 0, end quote. Author Bruce Fenton summed it up nicely, quote, "There is yet no known evolutionary mechanism or environmental forces that would bring about such changes. As we have discussed, the stability of these areas is essential to a healthy organism. Even with this glaring anomaly, we might be tempted to put it all down to just a strange freak event, until we learn that there are now several hundred human-specific accelerated regions of DNA code identified by scientists. While the vast majority of HARs remain mysterious in function, it is understood that they tend to modify the development of the fetus, and that most are not inside genes but rather the switches which control gene expression, modifying the degree of function or turning genes on and off. The researchers involved in the study of HARs suspect that it is these anomalous variations in highly conserved regions of code which brought about the most profound differences between humans and their closest primate relatives. Astonishingly, and beyond any reasonable coincidence, more than half of the genes located near HARs are involved in brain development and function. This does not look like a random scattering of sporadic mutations, not at all. With this understanding, we can look at the hundreds of anomalous human-specific accelerations which occurred after the split from chimps and recognize the handiwork of master geneticists, scientists, with an understanding that is light years beyond our own, and likely many light years from their home. When all known evolutionary mechanisms are factored in and the numbers are crunched, there is a statistical chance at zero that these HARs would exist. Yet, there they are. Fingerprints of the gods, a cosmic message in a biological bottle. While some will say that we can't prove aliens did this, I would counter that science does not work that way. We should accept the strongest model which best explains the observed evidence. Right now, the only scientific explanation that immediately explains all the HARs and many other anomalies specific to humans is the one in which an unnatural event occurred, and something overcame all of the mechanics of evolution which had preserved our essential genomic stability without killing us. We only know of one such force in the universe: intelligence." End quote. So, there you have it. These numerous discoveries in recent years have both bolstered and complicated the arguments of the scientist and the creationist alike. Both groups can find additional evidence to support their positions, but oftentimes the same new discovery used for support can also diminish their respective arguments. For those of you who have been researching the third option, which considers extraterrestrial intervention, you know well that both these groups can be characterized as partially correct to a certain extent. But neither group seems to want to acknowledge the potential identification of the missing link as being extraterrestrial. And this is quite ironic when it comes from the creationist crowd who believe a literal supernatural being breathed life into clay. The archive believes it is reasonable to state that the level of our current scientific understanding may be too limited to make absolute conclusions about our origin as a species. Is it really an evolutionary lucky roll of the dice that got us to this level of development? If so, that answer sure feels like a shallow scientific cop-out. Did extraterrestrials such as the Anunnaki intervene in the evolution of humans on Earth? Well, as far as speculative theories go, it cannot be proven any less valid than a lucky roll of the dice.